Good afternoon, everyone, and welcome to the second webinar in the Cancer Genomics online series. My name is Diana, and it's a pleasure to be introducing you to today's speakers. But before I do, I have a couple of quick announcements to make. First, I want to say a huge thank you to our series sponsors. Without their support, we would not be able to put this together for you. And I also want to say a huge thank you to our speakers, all of whom have joined us live today. So please do note the questions tab on the right. That's the best way to interact with our speakers throughout the webinar, and we will be doing an extended Q&A session at the end. So without further ado, I'm going to hand it over to our speakers to introduce themselves. Alice, would you like to go first?
Hi, everybody. My name's Alice Garrett. I'm a clinical genetics trainee by background, but I'm currently working as a clinical research fellow at the Institute of Cancer Research.
Thank you, Alice. Craig?
Yeah, my name's Craig Johnston. I lead the diagnostics and genomics team here at Automata. We are a lab automation company based in London, as you can see, flanked by some robots here. And I'm going to talk today about how to get your lab automation working smarter for you.
Thank you, Craig. And Alona, would you like to introduce yourself and kick us off with the presentation?
Thank you, Diana. Hi, I'm Alona Sasinski. I'm Scientific Director for Cancer at Genomics England. I've been in Genomics England almost for 7 years. Yeah, I don't quite remember what was live before that. Wait in my slides. I will talk today about integrating whole genome sequencing into the clinical practice for cancer patients. Yeah, I don't have a sequencer on my background. Greg does. Yeah, so this is what is Genomics England, and here is what we deliver. We have these 2 cycles. One is clinical, one is research, but as you can see, they're very closely connected. So I will focus for the next bit of the talk on how we integrate it into the genomic healthcare and how we delivering for the genomic healthcare. And the last bit of the talk, I will talk a bit about research and how these 2 things are connected. The idea is that all genomic information, all patient genomic information, of course, given patient consent, is deployed to the research environment, and academic scientists and pharma companies have access to this information to generate new insights that can be quickly translated back into the clinical space. So as I said, I will first concentrate on the patient journey, what our clinical cycle look like. So patients get recruited through the NHS genomic labs. Patient consent for clinical use and for the research is signed. Samples are collected, DNA is extracted, again in genomic labs, and sent to the Illumina Central Facility for sequencing. Once genomes are sequenced, they deliver to the Genomics England, where we do extensive quality control, mapping variant calling, and automated interpretation, prioritization of the variants in the patient genome. These results are returned back to the clinical scientists at genomic labs for more detailed interpretation, presentation to the Genomic Tumor Advisory Board, and then with expectation that these findings will make refinements, changes to the patient management. So whole genome sequencing is delivered now as a part of the NHS Genomic Medicine Service. You can find National Genomic Test Directory, which described all the tests that are delivered by the Genomic Medicine Service for specific indications by specified technologies. And on the left-hand side is the list of the clinical indications which are approved now for the whole genome sequencing. And I will mention a few of them later. So now we have this full-size healthcare ecosystem. As I said, National Test Directory is specified genomic tests that a patient can be eligible for. It's delivered through the 7 genomic lab hubs. We do invariant calling, interpretation in Genomics England, and then results are delivered to the GTAB. Unfortunately, I cannot go through the slides step by step. So On the right-hand side is what National Test Directory looks like. You can Google for the full list of tests. So for each clinical indication, it's defined what are the tests patient can be eligible for. And you can see that this tiny biopsy can be cut into the even more tiny pieces to deliver immunohistochemistry, different types of variants, tumor mutation burden, homologous recombination deficiency, FISH for some fusions, or RNA-seq panel for fusion. And we also have to do, for these special specific indications, we also need to do some search for the predisposition mutation in the germline. So for some clinical indications, this list is pretty long. So how we can, instead of running these multiple tests on this tiny biopsy, how we can potentially replace it with the whole genome sequencing and deliver everything with this one-stop shop concept. So this is just a couple of examples, and I deliberately focused here on pangenomic markers that we discover from the whole genome sequencing in addition to the mutations in the specific genes. So for example, this patient with high-grade glioma, whole genome sequencing was delivered. We ran signature analysis. It's on your top left. And we saw that patient came up with the specific pattern of mutations which usually associated with mismatch repair deficiency. And if you look at the mutation burden on the top right, you're probably familiar with this Alexandrov plot. So each column here is the specific clinical indication. With the red arrow, I'm showing adult gliomas. And on the y-axis is the mutational burden. So you can see that for glioma, there is a huge range of mutation burdens for these patients. But this specific patient also came up with ultra-high tumor mutation burden. You do expect high mutation burden for this patient with mismatch repair deficiency. But when we looked at the germline of this patient, we also find pathogenic stop gain in MSH6 gene. And also in our copy number analysis, we find that PMS2 gene was completely deleted. So all these pieces of puzzles put together, we are very confident that this patient have mismatch repair deficiency and very likely to respond well to the immunotherapy. This is another example, another glioblastoma patient. This is the recurrence of primary tumor, and actually clinicians were very surprised because this patient already survived 7 years past its diagnosis. So that's making super survival. If you look at the mutation burden, then again, it came up with very high mutation burden. Actually, it came up on the same GTAP as the previous case. So people on the GTAP said, oh, it's another case of the mismatch repair deficiency. But when we look at the signature analysis, we realize that actually this high mutation burden is just a consequence of chemotherapy treatment. And we also look at the distribution of variant allele frequencies for the SNVs in this patient. And we realize that most of these variants have very, very low VAF. So it's just these sporadic somatic mutations which emerge as a consequence of the several cycles of chemotherapy. Okay, so it's definitely not a patient with the mismatch repair deficiency. What else we can say? So we looked at our small variants prioritized in domain 1, and actually there were 50 variants in the genes that can be actionable due to the high mutational burden. So obviously most of them have low VAF, so they just pass passenger mutation. So we prioritize these 50 small variants by variant allele frequency. And there were very few with the high variant allele frequency. That means that they're present in each tumor cells. And oops, one of the top hits was RB1 loss of function deletion. And this explains why this patient is super survival. This is also make patient eligible for the clinical trial. It's very promising the clinical trial looking at Aurora kinase inhibitors. Let's move to the next one. Yeah, so now just a few slides on through our 100,000 genome project that probably Genomics England is best known for. We collected about 17,000 tumor normal pairs. So how this collection can influence future treatments, future changes to the patient management. So here we try to map the 17,000 tumors to the targets which are already in National Test Directory. So for example, as I mentioned, gliomas are already actionable. In each arrow in these heat maps are the different clinical indications, and each column are the different target genes. And on the left-hand side, we show genes with the highest prevalence of the small variants. Then the next, so I'm showing only 5 top genes. Then the next one is copy number changes, then structural variants, then homologous recombination signatures, mismatch repair signatures, tumor mutation burden, and then also prevalence of mutations in germline predisposition genes. So I highlighted here just 3 examples of clinical indications which are already eligible for the whole genome sequencing, and this is why they are eligible. So numbers in each of the cells showing prevalence of patients with a specific indication in these specific genes. So for example, for glioma patients at the top, you can see that they have high prevalence of the copy number changes in the genes which are already in the test directory. So very good target. Then the next one is ovarian cancer. And you can see that ovarian cancers have high prevalence of homologous recombination deficiency. This is a pan-cancer marker, which whole genome sequencing gives a high accuracy of identifying this marker. And you also can see that in ovarian cancer, there is a very high occurrence of the variants in the genes which predispose to the development of this cancer, especially BRCA1, BRCA2. And sarcoma is another indication which is already in the test directory for the whole genome sequencing. And you can see why. There is a high prevalence of copy number changes and structural variants in the genomes for this clinical indication that can be potentially actionable. So using these 7,000 genomes, which we sequenced so far, and mapping them to these actionable targets, inform commissioning of the new clinical indications for the whole genome sequencing. We also started looking at the potential prognostic markers. So, for example, KRAS was known to be prognostic marker for some like non-small cell lung cancers, for the metastatic colorectal cancer. So here we can look across different indications across the entire collection of our 17,000 genomes. And you can see that patients with the wild type KRAS have better prognosis. It's other way around for the PIK3CA. Patients with PIK3CA mutation have better prognosis. So here is the summary of what I was talking through. Whole genome sequencing is clinically accredited test delivered through the Genomic Medicine Services and National Test Directory in England. It's a one-stop shop for different types of the variants for the pangenomic markers. It gives you high resolution genome-wide capturing of the somatic mutations, germline mutations, and it provides you insights into the mechanism that drive the development of tumor. And the last but not least, we can use genomic data that we collected through the last 7 years, link it with longitudinal life course clinical data to accelerate implementation of the cutting-edge science into the clinical practice. Thank you. I'm handing here to the next speaker, and I see you later for the Q&A session. Thank you.
Thanks, Alona. That's over to me, and I will share my screen and we can kick off. Perfect. So yeah, as mentioned earlier, I lead the diagnostics and genomics team here at Automata. We don't actually have a long background in the world of labs. We started out back in 2015 as a robotics provider to largely manufacturing industries. But over the course of the past 7 years, we've realized the value of that manufacturing approach and that total automation approach, and that it can bring to the world of clinical labs. And the past couple of years have narrowed that focus, and I now work a lot with cancer genomics labs. That's where I spent my morning, got back just in time for this, reviewing some of their protocols. So that's what I want to run through today is really using my heritage and Automata's heritage in manufacturing to show you a new approach to lab automation within clinical diagnostics, and to just introduce you to some new ways of thinking, some ways to challenge some conventions in your lab to help your automation work smarter, and show you what we think the future of lab automation for cancer genomics is. And I guess the theme of today was clinical cancer genomics in 2022 and beyond. And I'll start with beyond. And what we believe the next generation of automation in cancer genomics will be is not going to be at an instrument level. We've been at SLAS, the Society for Lab Automation show, in the last couple of years, and there continues to be amazing advancements in lab automation at an instrument level. Liquid handlers get better and better year on year. Extraction platforms get better and better year on year. Every stage of a cancer genomics pipeline is really well served with an incredible instrument set. But fundamentally, these instruments don't meet the current challenges that we see day to day in cancer genomics labs. Whenever I speak to staff, be it new technicians on the floor, lab directors who've been in those facilities for years, the same challenges come up again and again. I don't have enough staff. I can't find any more of those even if I wanted to, because everyone wants those people. There's probably a lot of you on this call today here in high demand in labs across the world. I've yet to go into a lab where they feel like they have enough space. We work just down up the road from the Francis Crick. We've got a big lab automation installation going in their genomics lab. I cannot believe they can fit it in. Every single inch of that lab is utilized to its max capacity. And that's the same if you're in a world-leading research institute or in a small genomics center at a regional hospital. And the big one is that they, given all those constraints, need to scale throughput up to tenfold. We're all on this call today. We all understand the potential of cancer genomics to really shape the future of healthcare. We're seeing health systems start to cotton on to this, and the work that Genomics England done and ICR are doing is really pushing this forward. And we're really at the start of where the impact will be, but to really fully realize that impact, these labs need to scale up throughput without adding any more staff and without having much more space, which is a bit of a challenge. And then when we start to contextualize that within their future challenges, which is probably going to need to scale even more than that 10x as this takes off. And also the fact that this is a rapidly evolving industry where you need to account for emerging technologies or emerging techniques or emerging platforms. It was a matter of weeks ago that Ultima launched with claims of the $100 genome. These sorts of emerging technologies will only continue to come in the next few years. And we know labs want to be able to invest in platforms that can account for that. So that's why we believe that the next level of automation within these labs will not be within the instruments. because they don't serve these challenges. And when we actually look at the challenge of scaling throughput within those constraints, it's clear to us that actually the limiting factor is not the instruments, it's the flow of samples through the lab. And that's where that manufacturing approach comes in, and that's where our history working in FMCG and within automotive and within metals manufacturing starts to come in really, really handy. And what I want to run through today is a few principles that I've picked up from my time in manufacturing that can apply to your lab. As I say, challenge convention a bit, take a look at some processes a bit differently, and then show you where we want to take this. So this is, I guess, a very, very high level 100,000 foot view of what the sample flow in a typical cancer genomics lab might look like. It's what we see kind of day to day. There'll be sample prep, be it bloods, bone marrows, FFPE, or NIPT samples. They'll run through an extraction process, generally go into a pre-PCR library prep, post-PCR library prep, and then sequenced largely on Illumina or Thermo Fisher equipment, but we're increasingly seeing Oxford Nanopore and PacBio adopted as these longer read technologies continue to proliferate. And as I said, the challenge for these labs is not that they don't have the capacity to run each of these individual processes or indeed sets of processes. It's that samples do not flow smoothly between them, and that whole process doesn't work in harmony. So that's what we're going to run through today. The three things: lessons from manufacturing, approach to Katechia lab, and those opportunities for efficiency. Let's jump on in. So this is the kind of journey that car automation went on. And we're talking the last 150, 200 years. Started largely manually, just as cancer genomics processes started in translational research labs, moved through to partially automated steps where machines automated steps of processes, but not entire processes. And now you can go onto the factory floor of Tesla or Ford, and a car will go from start to finish without being touched by a single human hand. And cancer genomics feels like it's in the middle of that evolution at the moment. There's lots of great instruments, there's lots of great pieces of equipment that do partial steps, but don't do that full process. And human beings are still at the heart of everything that goes on. And that's what we want to bring in the future that we want to usher in is a much more high-tech cancer genomics lab. So these are the kind of four broad approaches that I want to run through today. And we'll go through specifically some examples of where you can find these sorts of benefits within a cancer genomics lab. The first is really an overarching principle, and that's to consider the holistic process. Often when we're working with labs on their automated processes, the first thing they give us is an SOP. And that SOP will handle part of a process, which is the best way to run an SOP. The best way to run a validation is to take the smallest component part of that process, validate it exhaustively, prove that it works, and then implement it into your clinical pathway. But when we're thinking about sample flow, we have to take the complete opposite perspective. And that's considering the holistic process. So considering the journey that a sample goes from start to end. And there's three principles below that, which we find provide the most throughput benefits to cancer genomics labs. The first is to optimize bottlenecks. There's a really easy heuristic, a really easy rule of thumb within the manufacturing world that we like to use, and that's your bottleneck should be on the piece of equipment that is your most expensive or most high leverage. You should always be trying to optimize or indeed maximize usage of your most expensive piece of equipment. If we're talking genomics labs, that's almost certainly a big, massive liquid handler, maybe a Hamilton, maybe a Beckman, maybe a TCAN system. And what we often find is when we go into these labs, those platforms aren't getting used enough. And that's because they are being bottlenecked by other processes around it. And I'll go into a bit more detail on that as we go. The second is to parallelize action. So again, we want to make sure that every single piece of equipment in that lab is working at once as much as possible. Often within cancer labs, we find that the extraction platforms run could actually achieve that 10x throughput that we talked about. But the problem is they're only being used for a certain set of the day. So understanding how you parallelize those in conjunction with your pre and post library preps and your sample preps, your sequencing as well, to make sure you maximize the use over an 8 or 12 or 16-hour shift, becomes incredibly powerful in terms of increasing throughput without actually increasing the amount of equipment, because remember, we have those space constraints that we talked about earlier. And the last is to provide connection. So in the car world, that comes from actually physically just moving the cars through these independent stations, which provide different services like attaching a bumper or painting a ar. In the world of FMCG, connections run through PLCs and conveyor belts, moving bottles between equipment in a beer factory, let's say. And in labs, the primary method of connection is through people. We'll talk about how we can change that a little bit later. But also adopting much more advanced LIMS platforms comes in here, much more advanced data tracking systems. Simply implementing a barcode scanning system can provide a massive amount of connection, which aids throughput. As soon as you take people away from manually exporting CSVs from, say, liquid handling setups, checking the data, reformatting them into a format, and then pushing them into a database, that person's time could be used much more effectively in that process. So it's these three things that we see again and again without investing in another major set of equipment or completely changing your space, can really provide massive throughput increases or massive increases in efficiency, which for a lab running 100 samples a week to a lab running 1,000 samples a week is valuable at any stage. So if we take that down a level and look at our kind of typical cancer genomics lab, that's the same slide again. Here's what we see the optimal way to optimize your bottlenecks. So as I say, your bottleneck should be on your most expensive, most high leverage piece of equipment, and that almost always will come within pre and post PCR library preps. Big chunky liquid handlers take up a lot of bench space, take up a lot of budget, and the closer you get those platforms to 100% utilization, the more efficient your lab is. And that's a really easy rule of thumb to understand how efficiently samples are running through your lab, is how much is my most expensive, my highest leverage, my big fancy liquid handler being used. Around that, there's some really easy wins. One is, say, if you're running this sample set, which was a kind of typical one we took from an average of a few hospitals we work with, we see a lot of manual blood prep. And people do that because it's easy for people to do, but actually those people could be used much better within that flow. So a really easy, quick win is putting in some basic sample preps for blood, making sure the bottleneck for your people is not sticking them on sample prep, it's utilizing them at higher value processes throughout that workflow. And then we often find that scaling the number of extraction platforms that you have before liquid handlers is usually a good way to go. And again, that's speaking in averages, so it depends on the lab. But as I say, we very rarely encounter a liquid handling or a library prep setup that's utilized as efficiently as it could be. So making sure that extraction is not your bottleneck or sample prep is not your bottleneck will massively increase the throughput of your lab without adding more massive expensive liquid handling setups. And if we talk about parallelizing, there's a couple of quick wins. So if we're talking in our library preps, if you're running a very long thermal cycle in the middle, which several protocols across the sequencing setup do run, then taking your thermocycler off the deck of a liquid handler can be a really great way to free up that liquid handler to run another library prep, but also make sure those pieces of equipment are working in tandem. We've watched processes where 300, 400, 500,000 pound pieces of equipment are just waiting for 2-3 hours while a thermocycling program runs. Those pieces of equipment could be much better utilized by taking that process off the deck and allowing those platforms to be used at what they are world class at. And then another quick win there is adding reagent dispensers to take prep work off deck. There's a really great range of very high speed reagent dispensers that exist out in the world that can prep a plate much faster than an 8-channel pipette head can in a high-spec liquid handler. And that can take a process that takes half an hour on a liquid handling setup down to 30 seconds, massively increasing the speed of that assay, but also freeing up half an hour on that liquid handling platform that can be better used. We're all about making things as efficient as they can be. And I talked about conveyors and FMCG or the platforms that move cars through factories. And for us, this is the future of a genomics lab. This is an end-to-end genomic setup that goes from sample preparation right through to sequencing without a human hand touching it. And this is fundamentally what we do. Automata brings these lab benches to market. We work with labs across the spectrum, primarily myself, as I say, in genomics and diagnostics, connect all the equipment together and help it sing in harmony. And we use those rules of optimizing bottlenecks and parallelizing actions to make sure we can maximize the throughput of these systems and really move to that world where people aren't moving microplate, or people with PhDs aren't moving microplates around a lab. They're there to analyze the outputs, and they're there to design protocols that are highly effective. And this is the level of connection that the genomics lab of beyond 2022 will have. And it's one that we're starting to install, as I say, at the Francis Crick, and we have a growing base of customers across the world who are adopting these bigger platforms where we can go from sample to sequence without a single hand touching anything in the middle, which for us solves the challenges that face the genomics lab and usher in that third pane that we saw in the world of car manufacturing that changed the industry. This is the way that we think that the world of cancer genomics is going to change. Thank you for listening to me. We'll wrap up there. Hopefully a few really basic insights that you can take back to your lab. If you are interested in learning a bit more about what we do, or probably more realistically understanding where these approaches could work in your lab, drop me an email. We're very happy to review some of your processes or just talk about how these approaches might work in a little bit more detail. 15 minutes is very quick to run through this big topic. So yeah, please do get in touch if you have any questions. And obviously we've got the platform to ask questions today. So yeah, thanks again for your time, and I will hand over to Alice.
Thanks very much, Craig. So as I said at the start, my name's Alice, and I'm a clinical research fellow in the translational cancer genetics team at the Institute of Cancer Research. And I'm going to change tack a little bit. So rather than talking about the sort of technological side of things, I'm going to talk about what I do, which is working on the interpretation of the results that come out of these amazing platforms and machines. So I'm going to talk about improving accuracy and consistency of variant interpretation for cancer susceptibility genes, particularly focusing on my work with the Cancer Variant Interpretation Group UK. And this work is part of the CanGene CanVar programme, which is funded by Cancer Research UK. So I know we've got quite a mixed audience. I'm going to do an extremely brief introduction to the field of clinical variant interpretation, and then, as I say, talk a bit about CanVar UK. I was hoping to tell you a bit about some examples of some analyses we've been doing, but I think I probably won't have time to cover that. So just to start off with, on the clinical variant interpretation side of things, anyone who has any experience whatsoever or has come within a whiff of getting involved in clinical variant interpretation will be familiar with this rainbow coloured grid published by Richards et al. In 2015 as part of the American College of Medical Genetics and the Association for Molecular Pathology guidance for clinical variant interpretation of sequence variants. And essentially the premise of this is that each of these little boxes has different types of evidence in it. So things like frequency and population databases, the outputs from computational tools, segregation or clinical data, things like that. And each piece of evidence is assigned a particular weighting. So supporting, moderate, strong, or very strong. And your evidence can either be towards pathogenicity or against pathogenicity. And then you look at the combination of bits of evidence you have of the particular strengths, and that gives you an overall classification of benign, likely benign, a variant of uncertain significance, likely pathogenic, or pathogenic. And in the original paper, they sort of set out particular combinations that you needed to achieve these different classifications. But most of the world is now moving towards a more sort of numerical points-based system, where you sort of can give your different evidence of different strengths, a number of points, and then your overall point sum will help you work out what classification to give your variant, as shown in this thermometer from the Association for Clinical Genomic Science. And as I say, this original 2015 paper was really, really helpful for improving lab consistency in variant classification, and trying to help people make sure they were doing similar things across different labs. But there were quite a few areas where perhaps it was a little subjective, or there was room for a little bit more clarification on different aspects of the guidance, be that particular evidence criterion, or perhaps particular genes or clinical contexts. And in this slide here, I've just got a few examples of publications that are out there relating to different parts of kind of building on that original guidance. So now moving on to the work that we do in the field of variant interpretation in cancer susceptibility genes. So the group that we lead at the Institute of Cancer Research is called CanVig UK, so the Cancer Variant Interpretation Group UK. And I'm not going to read all of these objectives out now, but I'll just very briefly go through each of them in turn and just give some examples of the sort of activities that we do in each of these areas. So to start off with, our first objective is to provide a multidisciplinary professional network and also a regular forum to discuss issues related to germline cancer susceptibility genetics. So the group has been running since 2015, and it was, and with our last count, we had 283 members from across the UK and also the Republic of Ireland. We have monthly virtual meetings where we do a sort of number of different things. So we develop our consensus specifications. We quite often discuss a tricky variant of the month, or a variant that illustrates a particular point. We sometimes have educational sessions, and also kind of relevant topical updates from various groups. And in between the meetings, we have a sort of email forum where people can discuss issues relating to variant interpretation that might be relevant to other members. And a number of years ago, we did a survey to sort of look at who our membership was. Obviously, we've grown a lot since then, but essentially about 50% of our members are clinical scientists, and the majority of the rest are clinicians, predominantly clinical geneticists, but we do have a few other people from other backgrounds as well. And people are doing a mixture of different bits of work. So some people predominantly cancer genetics or exclusively cancer genetics, but some people have a sort of broader range of different pieces of work. And this is just an example of one of our variants of the month. So essentially, before the meeting, we ask people to submit their variant classifications. So you can see the overall classifications and then the number of points that people were assigning to the variant. And then we encourage people to give comments about why they did or did not select different pieces of evidence to apply at different strengths. And these codes, these funny codes along the bottom, kind of correspond to different types of evidence as per that grid that I showed you at the start. So that allows us to see what types of evidence people are including, and that's really helpful for discussions during the meeting. So in terms of the training and education side of things, as I say, often at meetings we have presentations which are prepared by members, which cover a particular tricky or topical area, and then we upload these presentations to our website afterwards, so people have access to all the material. And we also contribute to other sort of educational endeavours. So on here I've just got a screenshot from a massive open online course developed by the team at St George's, University of London, who are also part of the CanGene CanVar programme on a different work package to do with medical education. And this MOOC just sort of teaches users about how the sort of premise of clinical variant interpretation, particularly relating to inherited cancer susceptibility in this case, and strongly building on the CanVig guidance. So another big piece of work that we do is we maintain a germline cancer variant specification. And as I say, this is building on the original framework, but just providing a little bit more detail on aspects which are relevant to cancer susceptibility genes. So just on the right there, you can see an example of what the guidance looked like. So it's, on some of these different types of evidence, it's got quite a lot of extra detail. And our original specification was published in the Journal of Medical Genetics in 2020. We've had a number of different iterations since then, and the latest update is available on our website as well. And this is just an example of one of the things that we've sort of added more recently since that original publication. And we've basically done a grid to sort of set out which bits of evidence can and cannot be used together, because there's quite a lot of overlap sometimes. So just to provide a little bit more explicit guidance on how to apply the specification. So as well as our main consensus specification, which is for sort of cancer susceptibility genes as a whole, we do also have additional interim guidance which we produced for particular genes. So we've got one, for example, for BRCA1 and BRCA2, and also for the genes which cause Lynch syndrome. And we also produce different pieces of guidance that we think is useful to the clinical community. So, for example, we've looked at a number of large functional studies in the literature and sort of assessed them and provided guidance about what strength of evidence these functional studies should be used at. And Miranda Durkee at the Sheffield Genomics Laboratory has worked very hard to produce a number of different report templates with suggested wording for reporting variants in cancer susceptibility genes that are used by a number of labs across the country to produce, to sort of decide what wording to include on their local report templates. So another large piece of work is the CanVar UK database, which is an online platform to help with information sharing and variant interpretation. And this is just a screenshot for it. So you can see sort of, it was very small, but it's basically got a lot of information about variants in the main cancer susceptibility genes, and links to various sort of useful databases like ClinVar and Varsome. And this website was originally worked on by Zhang Kut, who's now left the team and has now been taken on by Subin and Charlie. And we pull in lots of different bits of information. For example, you can see here a number of in silico tools, but we've also got a number of other bits of information. For example, we work with NHS Digital to pull in counts of variants that have been seen across labs in the UK. And this is really useful for case control analysis for variant interpretation, as we actually know the number of times a particular variant has been seen in cases referred to diagnostic laboratories, and we actually have unusually a denominator for the number of tests that have been done, and that's really useful for comparing to population databases such as NOMAD. And another way that the site is really useful is for discussion amongst different laboratory colleagues. So previously, as I say, we had this email forum where people would sort of send each other emails to the group saying, have you seen this variant? Have you got any further information? And now all of this is done through the Canvar site. So when someone has a query, an email like this will go into everyone's email accounts. And then when people reply, they can do this via the site as well. And the discussion is stored against the variant so people can see it in the future. And this feature is only available for logged in users, which is currently only people working in the NHS. But we have had a lot of interest from international users, and we're considering expanding internationally. And the last objective is our work with international variant interpretation endeavours. So we work closely with a number of different consortia, so for example the Enigma consortium, and we also publish our work in international journals, such as we've recently published this article on the reclassification of sequence variants in Genetics in Medicine, and Lucy Lung, who's another clinical research fellow in our team, presented this recently at the American College of Medical Genetics conference. Whoops. So as I say, I think I'm running low on time, so I won't talk in any detail about the analyses that we do within the group, but we have published a number of papers relating to essentially analyses which try and further specify how some of these evidence criteria could be used. So I've just highlighted in the red boxes some of the different criteria that we've covered. And the useful thing about that is that the evidence that we get from these analyses can then go directly into the guidance that we produce after ratification, obviously by the clinical and laboratory community. So it really, the impact can happen really rapidly, which is fantastic. So I will finish there. And I just wanted to say thank you to a few people who've been involved in this work. So particularly Beth Torr, our program manager at CanGene Canvar, and Professor Claire Turnbull, who leads Canva UK and the CanGene Canvar program, and who I've pinched a number of these slides from. Our team at the Institute of Cancer Research, our various national and international collaborators, the team at NHS Digital, our fantastic Canva UK membership group, and particularly our Canva UK steering and advisory group, CSTAG, who are a number of senior clinical scientists and clinicians who work tirelessly to make all this work happen. So thank you very much, and I'm happy to take any questions.
Thank you very much, Alice, for that presentation, and Ilona and Craig. If you could please join me on the stage, we will now do a few questions from the questions tab. This is just a last-minute reminder that if you do have any questions, please do put them in now. I have noticed Ilona and Craig have answered quite a few in there already. So a question for you, Alice, was do you have guidance and workshops for determining cancer driver mutations that are not necessarily germline?
So our area of work is almost entirely germline. We do sometimes get information from the somatic data, say for example about loss of heterozygosity and things, but our specifications are purely for the germline variants. There is another group, so we're CanVig, and there's another group called SVIG, who are currently working on somatic variant interpretation guidance.
Perfect, thank you. Just one for Craig. Does your work include automation of QC in the labs that's not directly related to the sequencing pipeline itself?
Yeah, absolutely. So the layout that we flashed up actually has 4 QC stages in it, and each of those we create daughter plates. So if any of the results of the QC or the sequencing, we need to recover samples, we don't have to go right back to the start. So we store those cooled at each stage of that QC step. So yeah, we tend to integrate a lot more QC when it's automated, because you have the ability to do so. So if that's a plate reader or a fragment analyzer, we tend to integrate a lot of QC into these setups.
Thank you very much. Alona, what is the method that you use for PMS2 copy number? What is the method used?
Yeah, at the moment, we are using alignments from Dragon, and we're using somatic variant callers from previous generation of Illumina pipeline. It's Strelka 2 for small variants, Canvas for copy number changes, Manta for structural rearrangements. We work now together with Dragon somatic Dragon team working on somatic callers with intention to completely Dragonize our mapping variant calling pipeline, because we find a lot of problems with calling copy number changes with Canvas, but at the same time, we need something with very quick turnaround times. All our pipeline takes more than 24 hours. So probably Dragon is where we're going to move next.
Super, thank you. That looks like all of the questions are currently answered. So while we're waiting for any additional to come in, I would love to ask your opinion, or all of your opinions, about what you foresee the future of cancer genomics taking place, because we've all got a different angle on this, with Craig being on the automation side, Alice on the kind of interpretation. From your individual opinions, what would you like to see moving forward? I'm going to start with Alice.
Well, I think there's loads of really, really exciting work that has been done in terms of, obviously the expansion of capacity and whole genome sequencing and things like that. But obviously I'm coming from the variant interpretation point of view, and all of those things are, they're limited by, your ability to actually understand what's coming up out of the bottom end. So I think, and a lot of the sort of the systems for sharing information and accuracy and consistency in variant interpretation are more primitive, like, seem to be kind of in a different world to the sort of fantastic robotics and things that, that you guys are talking about. So to be honest, focusing on some of the basics and is where I think the clinical impact is going to be really. Sorry, not very glamorous.
Alona, any additional comments to make on that?
I think workforce education is probably one of our main bottlenecks, because these genomes are really difficult to make interpretation of. So we need clinical scientists who can scale up their efforts and do it quickly. We're working now on decision support system to accelerate the process, facilitate, make it more simple and straightforward. Of course, price of genomes, hopefully, fingers crossed, will drop down soon. We all read this paper about $100 genome end of last month. So those are the things.
Craig?
Yeah, for me, it's always going to be around the volume that can go through these labs. So I think we're starting to see the GLHs in the UK set up, and we'd just love to see those be really supercharged over the next 2-3 years to make this accessible to everyone. I think the opportunities in healthcare are endless. So for us, it's just about scaling up capacity and letting the scientific teams do more science.
Thank you, Craig. And one final question. This is for Alona, and it's regarding access to EHR data from the 100,000 Genomes Project. Is this accessible to research teams, or is it only possible through collaboration?
No, of course it's available in our research environment. Actually, I was writing answers. I was going to copy paste link to how you can register for as our GCIP member. GCIP stands for Genomics England Clinical Interpretation Partners, but it's basically academic researchers who using our data. So all genomic data, all clinical data, patient electronic records, it all sits in our research environment. You can Google for the Genomics England GCIP and to find out how to register.
Thank you so much. So that is all of the questions that have come in. All of the questions were answered in the chat tab. So if anyone was interested in checking other questions that were asked, please do have a look there. But for now, that is the end of this webinar. Thank you so much, Alice, Alona, and Craig, for taking the time to present today, and for the audience for joining us live. We are live again at the same time next week for the final webinar in the series, so do mark your calendars. But in the meantime, thank you very much for joining. Have a lovely afternoon, and I look forward to seeing you in the next one. Bye-bye.
Thank you. Bye.
Bye.